So on the topic of ribosomes, it can be really informative to look at information processing in different organisms in an attempt to infer evolutionary history and relatedness between organisms. Information processing, in this case, refers to the set of tools and processes that different organisms use to take the genetic information stored in DNA and turn that into fully expressed and functional proteins. And so in particular, you have DNA polymerase that replicates the genome, you have RNA polymerase that converts DNA into RNA, and you have ribosomes that take RNA and use these RNA templates to produce proteins. And when you look at just the 3D structure of RNA polymerase in representative members of bacteria, archaea, and eukarya, it's pretty obvious that archaea and eukarya are more closely related to each other than they are to bacteria. You can see that all three domains seem to share a large portion of this structure, and yet you can see several of these domains are unique to eukarya and archaea. And so it doesn't make sense to say archaea and bacteria should be grouped together as prokaryotes when archaea and eukarya are more closely related to each other evolutionarily. If you do want to simplify things down into two branches, you need to come up with a new name for this branch of archaea and eukarya, as they share a common ancestor that they don't share with bacteria. And so this classically conceived prokaryote clade, it's not valid, it's not meaningful. And while these images here show RNA polymerase in particular, the same is true for ribosomes. If you look at the structure of the ribosomes in bacteria, archaea, and eukarya cluster together separately. So another way to look at the information processing abilities of different organisms, rather than looking at the 3D structure of RNA polymerase and DNA polymerase and ribosomes, you can look at the primary structure of these different kinds of things. And the primary structure refers to the actual sequence of amino acids, or the sequence of nucleotides in the case of nucleic acids, that fold into these secondary structures, which are these isolated domains, and then eventually fold into tertiary and quaternary structure, which is the 3D structure and how these fold in 3 dimensions. All of these are a result of the primary structure, and the primary structure gives us a way to infer relationships between organisms with a lot more resolution than looking at 3D structure. So here we have a super simplified diagram of some kind of chromosome, and somewhere on that chromosome we have a sequence of nucleotides that are aligned between different organisms. Now the fact that we're looking at a chromosome suggests that we're probably looking at in this case, this diagram is probably for humans, but the same goes for any organisms, in that all you have to do is find a sequence that is conserved between different life forms, and within that region, you look for variation. And so if you're looking at some conserved region that's found in all life, like some kind of information processing genes, you can look at these differences on the level of individual nucleotide substitutions in particular genes, or in particular stretches like you see here, or in particular stretches of this secondary structure. But you can also look at the entire genome. And when you start comparing these individual nucleotide differences across the entire genome, you can really start to see how life evolved from earlier ancestors to what we see today. And so the more different things are on the basis of individual nucleotides, the further they are from each other evolutionarily. And when you compare these findings to the findings of the information processing 3D structures, they overlap really well. And when you do that, you end up with, you can get a lot of different trees depending on how you evaluate the data and depending on which part of the genomes you're looking at. But on one hand, you can get a 3-domain tree, which I've described earlier, where you have eukaryotes and archaea sharing a common ancestor separate from bacteria. And so you have bacteria, you have archaea, and you have eukarya, with archaea and eukarya being more closely related. On the other hand, some more recent studies actually suggest this so-called eocyte tree, which shows that eukaryotes actually branched from within archaea, as opposed to simply sharing a common ancestor. And so there was this so-called eocyte at some point in the evolutionary history of archaea that turned into modern-day eukaryotes. Now, both of these trees are a lot more valid, I would say, than classifications based on the presence of a nucleus, or the presence of multicellularity, or any of these other kinds of relatively superficial features. Given the fact that we have this ability to look into the deep past and infer evolutionary relationships, it doesn't make sense for us not to do so. And the fact of the matter is, this three-domain tree of life was discovered about 40 years ago, as soon as this phylogenetic concept of inferring evolutionary relatedness by way of comparing genomic sequences, as soon as we started doing that in the 70s, we came up with this three-domain tree. We came up with this idea that prokaryote is not only not as useful as phylogenetics, it's at odds with phylogenetics, and it's not valid phylogenetically. And so, not only is the prokaryote idea 40 years out of date, even the three-domain tree is somewhat out of date. This eocyte hypothesis is the more currently accepted one. And so, overall, I would argue that the word prokaryote has far outlived its utility, and as much as possible, we should stop using it.